Okay, now you can start.
Okay, so my name is Anusha, and Can you hear me, right?
Hi.
Yes. Okay. So I just wanted to tell you all about brief about myself. That is, I'm into clinical SAS. I'm a SAS programmer from past 7 years. I'm working for a pharma company, and I'm into this industry from past 7 years. So from the clinical trial part, like the clinical operations, along with the what how the data flows from clinical operations, what happens in the site, and then from the site how the data is collected into the DM, that is data management. From DM, how SAS programmer, what is the need of a SAS programmer. So basically I know all this, like as I'm into this industry from long time. So today I'll be teaching you all just I'll give you some demo session so that you all will understand like what as you all know, like SAS can be it's nothing but a software which can be used either in a banking industry, it can be used in a clinical industry. It's just a software, right?
Sorry, Anusha. Sorry to interrupt you. Can you share your screen?
Yeah. I'll share my screen. Okay, just a second. So you're able to see my screen, right?
Actually, no. Just click that share button. One. Now we are start. Now it's starting.
Okay, so you can see my screen, right?
Yes.
Okay, so clinical SAS. Okay, so this is what we need to understand basics, like what is a clinical SAS, and then this is a content like for clinical SAS. What are the topics I'll be covering for clinical SAS? Okay, so first is okay, so this is nothing but CDISC, STTM, ADAM, and TLFs. Okay, so I'll tell in deeper view. So just first let's understand what is the introduction about the course, like what we need to know first. Okay, so first let's start with the introduction. So in introduction also there are like clinical operations. So clinical operations are nothing but trial, okay, on where the subjects are exposed. Okay, so clinical, I'll tell in the deeper like I'll explain it in detail, but just I'm understand making you all understand like, what are topics to be covered. Okay, and then like CDM, biostatistics, and medical writing, what this each department does. Okay, how we are related, how a SAS programmer is related to this particular department, how do we get the data. So that is about this. And then I'll be giving a more detail like what to say, a detailed view, like how the statistics, what is the statistics, and what are the we as a SAS programmers, what we need to do. All that will come under this part. Okay, and then the regulatory body, like what is a regulatory body and how the data has to be submitted to the regulatory body. At the end of the day, like whatever programming we are doing, it has to be approved from the regulatory body in order a drug to be released in the market. Okay, so just a introduction, like how, what are the regulatory bodies and all that. Okay, and then specification. So like I'll be showing you all the real time like I'll be giving you all the real time experience, like what is a protocol, how we need to write a program using a SAS. Okay, and then what is a specification. Specification is nothing but a document where we have to see the document and write the programming. So I'll go into a more deeper sections for this. Okay, and then the second part, it's like nothing but CDISC, SDTM, like what is CDISC, what is SDTM, what is like brief introduction, like what how the CDISC, what are the CDISC standards. Okay, so this and all will come under second part. Okay, and then the third is like IG, SDTM IG. Okay, so that IG, what are the fundamentals of SDTM. So why we already when we are having CDISC, already when we are having the data, why we need to follow this SDTM, what is SDTM. All this will come under this part, section 2. Okay, part 2. So I'll be covering all this, like what is SDTM, what are observations, variables, and then what are special purpose data sets, how do we do mapping, how do we do programming. Everything I'll be teaching in detail. Okay, and then submitting the data in a standard format. That's what I said, right? In like above I mentioned, right, regulatory body. So the data has to be reviewed by the regulatory bodies. So how do we submit the data to the regulatory bodies? So we have to have knowledge about that, like how the data is been submitted to the regulatory body. So this also I'll be explaining in a detailed view. Okay. And then these are the kind of domains I'll be showing it like in my next class probably, what are these domains, like how we need to map and all that. So these are the basic domains which we most widely use in clinical industry, and we have to have the knowledge about this. Okay. So these are the domains. So like how, what are interventions, events, findings. And then next, the SDTM mapping. Okay. So what is SDTM mapping? How do we use SAS in SDTM, like to write a program? So as you all know, right, SAS, like it's a programming language. So how do we implement that programming language into our clinical side? So that is what I'll be teaching here. And then SDTM annotations on CRF. So I'll be showing in detail like what is a CRF, how do we do and all that. And then SDTM mapping. So this is nothing but a I think I'll just show you. Okay, so it's just an introduction I'm giving. So this is nothing but a data, this is nothing but a project on which I am working currently. Okay. So you can see here, right? So as you can see here, there are set of lots of all this like tabs are there at the below. So these are nothing but all the domains. I'll tell in a more deeper view. So this is nothing but a specification. So this is a specification where we have to write the program. Seeing this specification, we have to write the program and generate one data set. Okay. And then later it will be proc compare will be done, and then it will be submitted in the form of tables to the FDA. So this is kind of just an example I gave. So this is the mapping. So this is the specification. So this, for this specification, there are certain rules how we have to do this. Okay. So this one I'll be teaching, and then later I'll be giving some real time project. Okay, like as I'm working right in the same domain, so I can give you my projects, real time projects. I'll give you some hands on experience, so that you all can get more hands on experience, and it will be easier for you once you get a job. Okay. And then later same thing for Adam. So SDTM, Adam and TLF, so three things are covered in clinical SAS. Okay. So now when you all will be thinking in your mind, already SDTM, the data has been submitted, so why Adam is coming into existence? Adam is nothing but a analysis. It's used only for analysis. Okay. Whereas SDTM is not used for analysis. It's just it's just writing the specification in the same format, writing the program in the same format. Okay. But for Adam, if there is any calculations or something, imputations, for all that things we are using Adam. So there is one more IG for that, Adam IG. So we'll be seeing that IG and we'll be writing the program. And then next is a TLFs. TLFs is nothing but representing in the form of tables. So T is tables, L is listings, and F is figures. Okay. So final data has to be submitted to FDA in the form of tables, listings, or figures. Okay. So reviewer, whoever is reviewing the data, he doesn't ask for any SAS data set or anything. It has to be uh, submitted in the form of a table, listing, or a figure. Like we are using lot of SAS procedures here, like for example proc frequency, proc means. Okay. And proc ANOVA, proc oncova, all these procedures will be used in this TLFs part. Okay. So just I'll be giving how to generate this TLFs and all. Okay. And then here also, see, I have mentioned here generating listings, generating reports, and generating graphs. So these are the nothing but basic things we have to understand in a clinical industry. So this is my content. Okay. So this I'll be covering, and I'll be giving real time projects on SDTM, Adam, and as well as TLFs. So you all will be getting hands-on experience on that. Along with that, I'll be teaching you all how to implement SAS in clinically, how we have to programmatically, how we have to derive all this. I'll be teaching. So let's get started now one by one. Okay. So let me tell you all briefly about this clinical terms, because I am not sure how many of you are aware of all this. So first is we'll start with clinical trial. Okay. So basics, we'll go with the basics. So I'll just okay. So what is the clinical research? This is very important for us to know. Okay. Before we start anything, we have to understand what is clinical research, which I was talking all this while. Okay. So let me, okay. So you all can see, right? So systematic investigation in human subjects for evaluating the safety and efficacy of any new drug. Okay, so this is very important. First we have to understand what is a clinical trial. So clinical trial is nothing but a systematic investigation in human subjects. So here human subjects are been have are for human subjects drugs are being given. Okay, and then why they are given to test the safety and efficacy. Okay, efficacy means how effective the drug is. So that is the systematic investigation. So that investigation is done in human subjects. So this investigation, whatever is done in human subjects, that is nothing but a clinical trial. Whenever an investigation is done on a clinical sorry, in a any human subject, that's nothing but called a clinical trial. So this is nothing but why it is done. Okay, second question. So it is done because mainly to bring out the new drugs into the market. So that is why a clinical trial is done, nothing but to get the new drug into the market. That's why the clinical trial is done. So now clinical trial, so for any trial there are some phases like phase 1, phase 2, phase 3, phase 4. So what are the like phases? Like it's done usually in 4 phases. Okay, I'll be explaining in a deeper section in next slide. So there are 4 phases in clinical trials: phase 1, phase 2, phase 3 and phase 4. So this all phases, the fourth like it's starting from the trials and then to the approval. So this all phases will be covering all this. Okay, so now see, it takes approximately 10 to 12 years and around USD 800 USD dollars. Okay, so one to get a one drug into the market, already it will take around this much of years, like around 10 to 12 years, whether it may be a small drug or a big drug. This is the minimum span for it. Okay, and then to get into the market, this many years it will take, and this many you have to invest this many dollars, like USD 800, that many dollars you have to invest. So it's a huge investment and it's a huge like time, right? So already if a drug has to be passed into the market, means the time span is huge. So trials have to be done accordingly so that the drug has to be launched into the market. Okay, so now see, for every 10,000 to 30,000 drug molecules screened, only one reaches the market. So molecules, okay, what is a molecule? Molecule is nothing but a drug. Okay, so molecule is a small like form in which the drug is investigation is drug done on the individual. So for every 10,000 to 30,000 drug molecules screened, only one reaches the market. So just imagine how much, like how much time and if you invest how many years and time or money, if only one will be reaching the market. So the trial has to be done in a very safe and efficient manner. So this is the very important part. Like what is a clinical trial? It's nothing but a systematic investigation in human subjects for evaluating the safety and efficacy of any new drug. So this is the main concept. This first one, first we have to understand. Okay, so now let me tell you all, just a second. um, okay, regulation. Okay, okay, fine. So next is, we'll understand what is the this is for clinical trial, right? Clinical operations. Now what is CDM? Okay, so what is CDM? CDM, I'll just in a detail I'll tell what is CDM, why we are. So okay, okay, so you can see right my screen. Okay, CDM. Okay, what is CDM? CDM is nothing but clinical data management. Okay, so CDM, after the clinical trial is done, for example, a trial is done on me. Okay, so the data, whatever the information, it has to be recorded and it has to be processed for further analysis. So that is why the CDM comes into existence. Because before how in before like in like any trial which has to be done, that has to be captured somewhere, right? It had the results. See, for example, today I'm given a drug morning. Okay, and suddenly my blood pressure raised, for about it was like 120 normal, automatically it became around 150. So the information, it has to be captured somewhere, it has to be stored somewhere, whatever that, after the information is after the drug is been given, the information has to be stored, right? So for that purpose, what they'll do is CDM came into existence. So it's nothing but methods and collections of data for purpose of clinical studies. Okay, that is nothing but called CDM. Okay, that's nothing but capturing the data, that's it. Or a technology and a process that manage clinical data to produce high quality, clean and analyzable database. So here, like in CDM, what will happen is there will be lot of databases. Okay, like I'll tell you all, electronic data capture, okay, EDC it's called. Like for example, Inform, Rave, all these are nothing but the softwares or the technologies or the methods in which the data gets stored in this form of databases. Okay, so the data will be stored in this databases, and then later what will happen, it will be sent to us, like for example SAS. So we have to start programming. Okay, so we'll see this. It captures the data into database, corrects the and validates the data. So this has to be taken care by the DM, that is the data management. The clinical data gathered at the investigator site in the case report form. So here what happens is, like there will be a form. Okay, it's just nothing but a form. Whereas case CRF means it's nothing but case report form, where where it captures, where the information about the subject, complete information, for example, name, age, sex, okay, all this, when he is ready, from when he started joining the trial. Okay, like all that information will be stored in this report form, case report form. I'll show you also how it is and all that, just you will get some idea. And then finally stored in the clinical trial data management system. So this is nothing but a CDMS, is nothing but a software where the data has been collected and stored about that particular subject. Okay, so this is nothing but the DM. So that's what I have written here. So clinical operations is, you know, right, the site, the where the trial has been done. And then the CDM. CDM is nothing but the data management, capturing the data. Okay. Now biostatistics. Okay, third one, biostatistics. What is statistics? I'm not sure, like you all know about statistics is nothing but it's nothing but gathering the collection of data in which is like it might be a demographic information. Demographic means of particular subject or on whom the trial is done. Okay, statistics is a science which deals with collection and organization, summarization, analysis, interpretation and presentation of data. So how do we present a data? That is nothing but a statistics. For what we do that, in order to get the interpretation and analysis. Okay, just to interpret something, some data should be interpreted. For example, you have seen this cricket matches and all, right? This data, how the data is interpreted in the form of graphs, in the form of charts, right? That's what in cricket it's like we have seen that, right, during the match. Same thing here. So why we do that is for easier understanding, like so that when a person sees the data, they'll easily understand like which drug is more effective, which subject was more treated very soon, kind of a thing. So that's why the statistics came into existence. Now what is biostatistics? This is nothing but statistics. Biostatistics is nothing but it is statistical methods and techniques applied to biological problems. Okay, biological problems or the data. Okay, so biological data means like, sorry, biological problems or data. See, that's nothing but the clinical trial. Okay, so in clinical trial what will happen is the trial on the first the trial is done, the drug is given. Then what will happen, how it flows is, the whatever the information they get, that will be stored in the EDCs, electronic data capture, I told, right, CDM. And then what they'll do is they'll send it, they'll send the data to SAS, and the SAS, we have to represent the data in the form of charts, or maybe in the form of tables, or maybe in the form of graphs. Okay, so that is how this biostatistics came into existence. Okay, now next is medical writing. Okay, so I'm not sure if you all know about this term medical writing. So it's nothing but it's producing the documentation of it's activity of producing scientific documentation by specialized writer who is typically not one of the scientists or doctors who perform or were involved in the research. So it's nothing but medical writers are persons who are not any of the scientists nor the doctors. Okay, they are not even involved in the research. So their main goal is to produce a scientific documentation. So they will be documenting scientifically, but they are not doctors nor any scientists. So that is their profession. So what they'll do is they have some they use some dictionaries, like for example medra dictionary. All the dictionaries they'll use, and they'll represent the data in a scientific documentation. Okay, they'll try to use more standard terms which is used by the WHO, who, or maybe medra dictionaries. So all these dictionaries will come into they'll just analyze that and they'll write a document. Okay, so this medical writing, it's a separate department actually. So these are the people who are who are specialized only in the writing using some specific dictionaries. Okay, fine. So any doubts you all are having till now, you all didn't understand, or you want me to repeat anything? So it's fine,
No,
right?
it's
Yeah,
fine.
you,
It's
Yes.
okay.
yeah, or
Bali, any
am I
query?
going?
No, nothing.
Okay, so am I going too fast, or maybe you can tell me, you can ask me to slow down, because maybe first, like I might be some fast or something. Okay, so these are the nothing but the just introduction I gave. Okay, so clinical operations, how it works, CDM, biostatistics, and medical writing. Okay, now,
Okay.
uh, now I'll tell you all what is SAS. Like, okay, I'll show you this slide probably. Okay, okay, so I'll just show you this. Okay, so now let's start with data management, because clinical research already it will be taken care by the site people. Site. Okay, one more thing. So now this, you might be thinking, who is doing this? Like, who is performing all this? Like site, who is giving this, all these drugs and all that? Okay, so the drugs are given by the doctors. They are called as physical investigators in clinical research terms. Okay, and on whom the subject, on whom the trial is done, they are called as subjects. Okay, so we do not in clinical trial we don't use any names, or we don't use anything, just we call them as subjects. And on the trials, on whom who are performing the trials, they are nothing but the physical investigators or doctors. Okay, so let's start with this now. So this is the clinical data management, how the data flows. I'll just explain you. Okay, so this is a protocol. Okay, I think I'm having protocol. I'll just show you. Uh, I'll just show you the protocol. One second. Yeah. Uh, yeah. This is a protocol. So these are the live things I'm showing. So this was a study which I worked. Okay, so protocol. Okay, so what is a protocol? Protocol is nothing but a document. Okay, so document which consists complete information of the study. Okay, it has complete information. Where is the like, for example, if you take the first page. Okay, so now this is the title. Okay, clinical investigation evaluating the efficacy. I told you, right? It's nothing but effective, how effective the drug is, of full thickness placental allograft in lumbar microdiscectomy outcome. So this is the title of the protocol. So primary investigator or physical investigator, he is nothing but a doctor. So this is his name. Okay, and this is the site. Site means, site is nothing but hospital. Okay, so that's what we use in clinical terms. So and then this is his address, phone number. So this is nothing but a protocol. So sponsor. Now who is a sponsor? Sponsor is someone who is giving money in order to conduct the trial. Okay, so this is the lab, stim labs. So this stim labs is providing money to conduct this trial. Okay, so this was a protocol which I worked. Okay, and this is the phone number and all these facts. Okay, so now let's see, this is the complete information of the study. So this is the, I told you, right, this is the title, and then these are the terms, how the study has been done. This is the duration, like how many years it exactly it has to conduct, like 2.5 years or this one. Study centers, multi-center means nothing but where it is where the trial is conducting in more than one center, more than one hospital. Okay, like for example you are staying for the, for example it's conducting in X, and one more site it might be a hospital, it might be conducted. So multi-center means many centers. Okay, you can see this, right? Polaris Spine, neurosurgery, Atlanta. So this is the study which I worked. Okay, and then in the protocol there will be some objectives. What is the primary objective and what is the secondary objective? So this is a very important question. Okay, when you are interviewed, they will ask this: what is a protocol? First question. Okay, second question: what is a study which you have worked? It might be internship or dummy project, whatever. So this title you should know. Okay, and then they will ask what are the objectives. So you should know this. Okay, this is very important in interview point of view. Okay, and one more thing is I'll be preparing you all for interviews also, and I'll be referring you all for the jobs also, because I'm having lot of references and I've placed many students also. Okay, so I'll be training you all and referring at and this what I'll make you all understand this by giving the project and giving some real time experience. Okay, for SDTA madam or TLF or all the three. So these are the primary objectives, is nothing but to evaluate the efficacy of Revita is nothing but a drug. Okay, and then so how effective this drug is on thickness placental allograft, on improving the back and leg pain post micro. So it's nothing but for a leg and back pain. So this drug, how the effective is, how effective it is. So that's what its primary objective. Okay, and then this is the post micro. Okay, so the secondary objective is to evaluate the post micro discectomy rehydration rate in patients with treated. Yeah, so this is nothing but the secondary objective. Primary objective is how effective it is. Okay, we have to check that, like some there will be some like statistical analysis for that. They will statisticians will decide like how effective the drug is. There will be some calculations done, like sample size calculations. Okay, so that molecule, how it's affecting, how many adverse events are happening. Okay, so if anything seriousness, very seriousness, or maybe some subject while performing the trial he had faced some adverse events, so immediately they will try to discontinue that drug. Okay, so and then see number of subjects. So number of subjects, how many are there? 182 subjects are there. Okay, and they are randomized into two arms. Okay, so what is the randomization? Okay, so randomization is nothing but in clinical term I can say it's given randomly. Drugs are given randomly to the subject so that there should not be any bias. Okay, so that is nothing but randomization. And then arms are nothing but treatment. Okay, what is a treatment? So treatment and control. So there will be two types of drugs. Okay, it depends on the each study. Okay, so this one it's like the main drug. Okay, and with the active ingredient, and the second one is the placebo without the active ingredient. It's just a dummy drug. Okay, so they are checking the effectiveness, that efficacy, taking the drug which has having the active ingredient, and other one which is not having the active ingredient. They're trying to evaluate that. Okay, and then inclusion and exclusion criteria. So what are the inclusion criteria? So there will be some inclusion criteria. Okay, like there should be some male or female with that particular age, and he should not have any some medical disorder. So that whole will be mentioned in this inclusion criteria. And then the exclusion, so there will be some, see they have written here, right? Smoking, additional health risk factors. They just don't prefer such people, so that they'll be knowing like how the drug is interacting with our body system. So they'll have some specific rules, like smokers and like some other risk factors they won't uh, appreciate all such things. So that is described under exclusion criteria. Okay, and then this study protocol dose, that's what it will be designed. It will be designed like it will be given like how much dose they have to take, and then what is the route means how he has to take, whether he has to swallow that or he has to take by injections. Okay, so this and all will be mentioned in the protocol. It's like complete information will be mentioned about this study. And then see primary endpoint and secondary endpoint. So these are the also important questions they'll ask. What is the primary objective? What's the primary endpoint in your protocol, sorry, in your study. Okay, so you can tell them, you can take this example and you can tell them lower back and leg pain is measured by the this ODI, and then second endpoint rehydration rate. So you can just tell them, so they'll understand that you have done some project. Okay, so after that, after all this, they'll be writing in a very deeper section, like exactly they'll explain everything and they'll write this. Okay, so all this will come into existence. So this is nothing but a protocol. Okay, so protocol is nothing but a document which gives complete information about the clinical trial. Okay, if they ask in interview, you can tell this, and you can just make a note of primary and secondary objectives. Okay, so now next, okay, so let me tell something about, before I proceed, let me tell you all something about, yeah, so here I was explaining, right? So this is nothing but protocol. I said, I showed you, right? One dummy, it's not dummy, it's a real time protocol. And then the CRF. Okay, what is a CRF? CRF is nothing but a case report form. Okay, so this case report form is nothing but like, I'll just show you. Okay, one second. Okay, okay, so the first one is the protocol, just I just showed you all, right? The second one is, this is nothing but the case report form. Okay, so it's nothing but a form which has complete information about the subject's name, sex, age, if he is having any other disorders. Okay, something like some other, like blood pressure or some other like diabetes, and if he is having some other related health disorders, everything will be mentioned. This is nothing but the case report form. Okay, so it is nothing but a set of a document, like for example, if you're going to any college for admission, so in that case, like just you want to give information to the college, right? Like my name, my father's name, my mother's name, all that. So it's that only, it's just a document which consists of the information about the subject. Okay, so one more thing, yeah. So it's in a, if you want the exact definition, means this is how it is. It's nothing but a printed, optical or electronic. Okay, designed to collect the data that is described in the protocol for each trial subject. For each subject, this is nothing but a document which gives information about the each trial and about the each subject. Okay, and then one more thing is, okay, so yeah, one more thing is, there are two types of CRF. Okay, one is nothing but electronic, directly it's captured, means it's directly entered into the system. Okay, one more is writing in a paper. Okay, so this paper format was before it was used, okay, in the sites. Now almost all the sites, they don't appreciate paper, they just go directly for electronic capturing, means directly entering into the system. Okay, that is what now all are following. Okay, so and then uh, just a second, yeah. Okay, so then another form is nothing but tables. Okay, so tables means nothing, this information is just, it will give the counts, like, oh, represent, that's what I told, representing in the form of a table, counts, like in the form of tables or in the form of sorry, graphs, bar graphs or charts. Okay, so just to make the reviewer understand in a simple way. Okay, so that is because the reviewer won't know the SAS programming, right? He won't understand SAS. If you tell them this many subjects are there, it's very difficult for him to see the data set also, he won't be able to understand. So that's why we submit the data in the graphs or charts, okay, in that form. Okay, so now next, just a second, I covered, right? Okay, so this is what, and next we'll see something about regulatory bodies. Okay, this is also very important to understand before we proceed further. uh, One second.
Hello,
Regular
sorry to
track.
interrupt you, Nisha. 4 minutes left only. So in the next
Okay.
session, we if
Yeah.
I think for the demo you can start in the next class also. You completed
Okay.
or not?
Fine. Yeah, yeah, it's completed. Just only one part was the regulatory. It's okay. I can take in the next class.
Okay, okay.
Okay.
So yeah, no problem.
Okay. So just let me know, like, if they're having any doubts, you can reach out to me.
I
Anything
think
as far as.
Dipali has want to ask something.
Yeah, yeah, Deepali.
Thanks, Anusha, for sharing the idea about, means knowledge about this clinical science. I got
Mm-hmm.
some brief idea. So I have
Mm-hmm.
some question type I want to share with you.
Okay.
That I'm in working for 4.5 years in the medical coding domain,
Mm.
being having a
Okay.
master in the biotech. So I want to
Okay.
enter in the SaaS domain. So for what it means scope for the SaaS, yes, I'm going to change my domain. Mm.
Okay. So you said you are into 4.5 years, right?
Yes. Mm.
Okay. So you know SAS, right? I mean, base and advanced. You have finished the course, right? Or you are still
Yes,
doing
yes.
the course?
Yes, I'm
Uh,
doing.
Okay, see, scope is for like it's in a wider range. Okay, I can
Mm-hmm.
say, because as already you are into clinical, like clinical in the sense not fully, but something related to clinical, right? It's not completely into IT like Java, .NET or something, right? So you are
Yes.
into clinical. So medical coding is like from medical coding, you are saying you want to move into clinical science. That's what you said,
Yes.
right?
Yes,
Yeah,
Asha.
so yeah, the scope is in a wider range, I can say, because see,
Mm-hmm.
for a for a clinical SAS, okay,
Mm-hmm.
usually there are very few opportunities for as a fresher, okay, to be very honest with you. But if
Uh-huh.
you do this
Mm-hmm.
course, and if you get some, like, if you understand this course, and if you do some projects, okay, so
Mm-hmm.
that what you can say is like medical coding I was also doing supporting for clinical, because that's what it is, like you're doing project, right? You can say I have worked on some projects, and then
Uh-huh.
it will be easier for you to get into SAS industry, okay, into clinical
Okay.
SAS industry. Okay,
Okay.
that is one thing. Second thing is, you, for example, your package is something, just example, 5 lakhs, just example I'm giving, okay. Maybe
Mm-hmm.
it's 5 lakhs. So you will be getting a good package, like around 15-20% compared to like current package of yours, okay.
Okay.
And third thing is you should be understanding thoroughly about this clinical SAS, like what is SDTM, what is ADAM, what is TLF. You should be thorough in programming part. That is very important, because programmatically SAS knowledge is very important to do. See, for example, I showed you a specification, right, just now in the
Yes.
class. This,
Yes.
see,
Yes.
now this specification, it looks simple,
Mm-hmm.
but when it comes to Adam specification, there are some analysis part and all that. So you have to be very strong in SAS, okay.
Okay.
And this like there are many domains in SDTM, so you can, it's easy, it's not that difficult. I can help you out, and I can refer you also, like after you, probably if you complete the course, I can refer you with my reference. You can get some, because I have placed lot of students, okay.
Okay.
So but moving to coder to clinical science, that's the best choice, I can say. And
Mm-hmm.
a lot of opportunities also there for clinical SAS nowadays. Everywhere SAS is in demand, even in abroad. You go to abroad or you go to India, wherever it is. So lot of scope is there for clinical SAS, okay. So that is one of the best decision you can take. You can try to understand things. I'll be giving you projects, I'll be preparing you for interview also, and after the course I'll refer you also, okay. So
Mm-hmm.
that is what I can say.
Okay. And
Hmm.
how is about the growth in the and what are the